Every telescope we ever built pointed at the wrong kind of planet. For 60 years, we searched for worlds that looked like Earth. Same size, same distance from a star, same liquid water. We assumed life needed what we needed. We were wrong. Life on our own planet grows inside volcanoes, survives nuclear radiation, and breathes chemicals that would kill us in seconds. We just stopped looking for it. Somewhere out there, a moon is shooting liquid into space right now. Another world has oceans that have been warm for longer than Earth has existed, and a gas 40 times deadlier than bleach is floating in the sky of our nearest neighbor. If you enjoy what we do here, subscribe and drop a like. It helps more than you know. 33 stops. One terrifying conclusion. We begin. Picture this. A room full of the smartest people on the planet, gathered in 1961 to answer one question. Where should we look for alien life? They pulled out equations. They drew diagrams. They argued for hours. And at the end of it all, they pointed at the sky and said, "Look for another Earth." That meeting happened at a research institute in West Virginia, and the decision made there quietly shaped every space mission, every telescope, and every dollar spent on the search for alien life for the next 6 decades. The logic seemed obvious. Earth has life. Earth has liquid water. Earth orbits at just the right distance from the Sunday. So find a planet like that, and you find life. Simple, clean, completely reasonable, and almost certainly one of the biggest scientific mistakes in human history. Here is what they missed. At the exact same moment those scientists were drawing their diagrams, living things were thriving in places no one thought possible. Creatures were eating sulfur in boiling hot springs. Microbes were reproducing inside solid rock, miles underground, with no sunlight and no oxygen. Life was doing things that broke every rule those scientists had written down. Nobody paid attention. Those discoveries felt like weird footnotes. The main search kept going, aimed at planets that looked like ours. Scientists even built a name for the target zone. They called it the habitable zone. It described the narrow belt around a star, where temperatures allow liquid water to sit on a planet's surface. Close enough to stay warm, far enough to stay cool. Earth sits right in the middle of ours. The habitable zone became the Holy Grail. If a planet fell inside it, telescopes pointed at it. If a planet fell outside it, scientists moved on. Decades passed. Billions of dollars flowed into instruments built to find Earth-like worlds. The Kepler Space Telescope alone discovered over 2,700 planets. Scientists celebrated every time one landed in the habitable zone. But here is where the story breaks open. While all those telescopes were scanning sunlit surfaces, something strange was being discovered much closer to home. Scientists found entire ecosystems living in places that should, by every rule we had written, be completely dead. A cave sealed off from the rest of the planet for millions of years. The inside of a nuclear reactor. The frozen surface of Antarctica. Each discovery chipped away at the same assumption that life needs conditions similar to ours. By the time most scientists started taking these findings seriously, the search for alien life had already baked in 60 years of Earth-shaped thinking. The telescopes were built. The missions were planned. The funding was allocated. Changing direction meant admitting that the entire framework had been too narrow from the start. Some scientists did admit it, and what they found when they started looking differently was staggering. Liquid water on the surface of a planet, it turns out, is one of the rarest setups in the universe. Most water exists underground, under ice, or inside moons that orbit far beyond any habitable zone. If life only grows where we expected, then the universe is nearly empty. But if life grows the way Earth keeps showing us it can, the universe is overflowing. That switch in thinking, small on paper, enormous in impact, is what the rest of this journey is about. And the first crack in the old model came from the last place anyone expected. A cave, sealed shut for 5 million years, with no light, no oxygen, and no connection to the surface world above it. What scientists found inside changed everything. One star. That is all it took to build a rule that governed 60 years of science. The habitable zone was designed around our Sunday scientists, measured how far a planet needed to be to keep water liquid on its surface, based on how much heat our sun produces. They found the sweet spot, somewhere between about 93 million and 150 million miles out, and they called it the Goldilocks zone. Earth sits inside it, Mars is right at the edge, Venus is just inside it on the other side. The math was solid. The problem was that scientists then did something quietly dangerous. They exported that math to every other star in the galaxy. Different stars run at different temperatures. A red dwarf star, the most common type in the universe, burns much cooler than our Sunday. Its habitable zone sits much closer in. A planet in that zone gets blasted with flares and radiation that our sun never throws at Earth. A massive blue star burns so hot that its habitable zone sits much farther out, and the star itself explodes after only a few million years, far too short a time for complex life to develop. Scientists knew all of this. They adjusted the math for each star type, but the core assumption stayed locked in place. The surface of a rocky planet, sitting in the right temperature band, was the target. That assumption eliminated most of the universe before the search even started. Consider what got ruled out. Gas giants were crossed off the list immediately, even though some of their moons have more liquid water than all of Earth's oceans combined. Icy moons were ignored for decades because their surfaces sit millions of miles outside any habitable zone. Rogue planets, worlds with no star at all, were not even worth discussing. The funding followed the assumption. Space agencies around the world poured resources into planet-hunting telescopes designed to find rocky worlds at the right distance. The instruments were extraordinary. The Kepler mission, launched in 2009, stared at one small patch of sky for 4 years and found thousands of candidate planets. Its successor, the Transiting Exoplanet Survey satellite, expanded that work across the whole sky. Both missions were brilliant. Both were aimed almost entirely at finding Earth. Scientists who argued for looking elsewhere faced a quiet kind of professional pressure. Funding panels wanted results tied to known life-friendly conditions. Papers about subsurface oceans on icy moons sat in journals for years before anyone paid serious attention. Then something happened that forced the conversation open. A moon of Jupiter, already known but barely studied, started showing signs that no one could explain away. Its surface was cracking, its ice was moving, something underneath was churning with heat. Scientists had to pay attention. What they found underneath that ice rewrote the entire search. But before we get there, something else happened first. Something that should have changed the game decades earlier and almost got ignored completely. Scientists cracked open a sealed cave in Romania, a cave cut off from the surface world for 5 million years, expecting to find nothing alive inside. What they found was a full ecosystem. A scientist named Thomas Gold spent years trying to convince his colleagues that life was hiding deep underground, miles below the surface, in places with no light and no oxygen, and temperatures hot enough to cook food. They laughed at him. Gold published his idea in 1992. He called it the deep hot biosphere. He argued that microbes lived in the pores of underground rock, eating hydrogen gas that seeped up from inside the Earth, completely cut off from the surface world above. He estimated that the total weight of all underground life might actually rival the total weight of all life on the surface. His colleagues thought he had gone too far. The idea was too strange. Life needed sunlight. Life needed oxygen. That was the foundation. Then the samples started coming back. Drilling teams boring down into ancient rock for oil and mineral surveys began pulling up cores that contained microbes. Tiny living things, locked inside solid rock, reproducing so slowly that a single generation took thousands of years. Scientists found them at depths of nearly 2 miles, then 3 miles, then deeper still. These organisms ate iron, they ate sulfur, Some of them breathed carbon dioxide instead of oxygen. None of them had ever seen sunlight. None of them needed it. Scientists gave this category of life a name: extremophiles, organisms that thrive in extreme conditions. And the more they looked, the more extremophiles they found. They found bacteria living in the cooling water of nuclear reactors, absorbing radiation levels that would kill a human in minutes. They found microbes encased in Antarctic ice, frozen solid for hundreds of thousands of years, reviving when scientists thawed them out. They found living things in the boiling acid pools of Yellowstone, in the hyper-salty waters of the Dead Sea, inside clouds of sulfuric acid. Every single one of those environments had been called a dead zone at some point in scientific history. Here is why this matters so much. Every time scientists found life in a new extreme, they had to expand their picture of where alien life could survive. A planet with boiling acid rain? Maybe fine. A moon buried in ice with no sunlight reaching the surface? Maybe fine. A world orbiting a dying star, bathed in radiation? Maybe fine. The definition of habitable kept growing, and as it grew, the list of worlds worth studying grew with it. Gould never fully got his credit. He died in 2004, a year before deep underground life became accepted science. A team using a South African gold mine drilled down over 2 miles and found microbial communities that had been living in isolation for millions of years, eating hydrogen produced by radioactive decay in the surrounding rock. Gould had been right. The implications hit scientists like cold water. If life on Earth already covers a range this wide, the question changes completely. The question becomes, what environment can life absolutely not survive in? And every year, that list gets shorter. One discovery, more than any other, forced the change. Scientists were not drilling deep underground when they found it. They were on a submarine, descending into the deepest, darkest, most lifeless part of the ocean. What they found down there changed the rules permanently. A highway crew in Romania was laying a road in 1986 when their equipment punched through the ground and opened a hole. Cool air rushed out. Someone lowered a light. Nobody expected what they saw. The cave below was enormous. The walls were coated in strange, pale formations. The air smelled of sulfur, and when scientists arrived and began carefully moving through the tunnels, they found animals. Dozens of species. Spiders, centipedes, water scorpions, leeches, and more than 30 species that had never been seen anywhere else on Earth. All of them blind, all of them colorless, all of them completely cut off from the surface world for an estimated 5 million years. Scientists named the cave Movile, and what made it more remarkable than the animals themselves was the chemistry keeping it alive. The cave had no connection to the food chains above. No sunlight reached it. No dead leaves fell in. No surface water flowed through it. Instead, at the bottom of a small lake inside the cave, microbes were pulling energy directly out of hydrogen sulfide gas, a toxic chemical that smells like rotten eggs and kills most living things. Those microbes formed a floating mat on the water's surface. Everything else in the cave ate them. The entire food chain ran on chemistry, with no sunlight required anywhere in the chain. Scientists called the process chemosynthesis, and Movile Cave was one of the clearest examples of it ever found on land. Life had taken a completely different path. Same basic result: organisms eating, reproducing, and dying, but powered by an energy source that no surface creature uses. The animals inside had been evolving in isolation for so long that they had lost their eyes entirely. Eyes cost energy to build and maintain. In total darkness, they serve no purpose. Evolution stripped them away over millions of years and used those resources elsewhere. Some species inside Movile have no counterpart anywhere on the surface. They filled roles in that sealed-off world that surface animals fill above. Predators, scavengers, grazers. A complete, balanced ecosystem running on toxic gas, in total darkness, completely sealed from the rest of the planet. The discovery rearranged how scientists thought about what a habitable world actually requires. A planet could have no sunlight reaching its surface. It could be wrapped in toxic gas. It could have its oceans sealed under miles of ice. And based on what Movile showed, life could still run a complete and complex food chain inside it. Within a few years of the Movile discovery, scientists began applying that logic to the moons of the outer solar system. Europa, Enceladus, and others had long been dismissed as frozen and dead. Movile showed that frozen and dark was not a deal-breaker, but the cave was just one piece of the puzzle. Scientists already had another discovery sitting in their archives, one made nearly a decade earlier in the ocean, that pointed toward the same conclusion and had somehow not yet changed the way the search for alien life was run. That discovery came from the bottom of the Pacific Ocean in one of the most extreme places on Earth, and the creatures found there should have ended the old assumptions on the spot. In February of 1977, a small submarine named Alvin descended nearly 1.5 miles into the Pacific Ocean near the Galapagos Islands. The scientists inside expected to find cold, dark, empty seafloor. They had their cameras ready to film nothing. Then the water temperature spiked. Through the porthole, the seafloor appeared, and covering it, in dense, colorful clusters around cracks in the rock, were organisms that had no business being there. Tube worms, some as tall as a grown adult, waving in the current, clusters of clams the size of footballs, crabs picking their way through the crowd, mats of white bacteria stretching across the rock, an entire thriving community, miles from any sunlight, in water, cold enough and pressurized enough to crush most living things. The heat came from below. Cracks in the ocean floor, called hydrothermal vents, were pumping superheated water rich in minerals and chemicals straight up from inside the Earth. The bacteria at the base of that food chain did the same thing the Movile microbes would be found doing a decade later. They pulled energy out of the chemistry. No sunlight, no photosynthesis, just chemical reactions. The scientists in Alvin radioed the surface. Their colleagues could not believe what they were hearing. For over 3 billion years, science had assumed that all life on Earth traced back to sunlight. Every food chain ended at a plant or algae that caught solar energy. The rules of life on Earth were written around that fact. The vents broke the rule completely. Here is where the story gets uncomfortable. Scientists understood the significance immediately. Papers were published. The discovery made headlines. And yet, for almost 2 decades, the search for alien life kept pointing at sunlit planets in habitable zones. The logic that should have followed, that life needs energy, and chemical energy works just as well as solar energy, took a remarkably long time to fully land. Part of the reason was how science is organized. Different teams study different things. The scientists discovering hydrothermal vents worked in ocean biology. The scientists searching for alien life worked in astrophysics. The two fields barely talked. Ideas that should have crossed over quickly sat in separate journals, read by separate people. It took a moon of Jupiter to force the conversation. In the 1990s, the Galileo spacecraft began sending back images of Europa, one of Jupiter's 4 large moons. The surface was ice, cracked, streaked, and shifted ice, with almost no craters. That lack of craters meant the surface was young, geologically speaking. Something underneath was churning it. Scientists looked at the vent ecosystems. They looked at Europa. They made the connection. If a moon had a liquid ocean under its ice, heated from below by tidal forces from Jupiter's massive gravity, and if chemical vents existed on the floor of that ocean, then the rules established at the Galapagos vents applied there too. Life could run on chemistry, in the dark, far from any sun. That idea shifted the entire search. And the more closely scientists studied Europa, the more extraordinary it became. What they found was an ocean bigger than all of Earth's oceans combined, sitting under miles of ice, possibly warm at the bottom, and possibly churning with exactly the right chemistry. The ice is about 10 miles thick in most places. Below it sits a liquid ocean. Scientists estimate it holds about twice as much liquid water as every ocean, lake, and river on Earth combined. That alone is staggering. A moon, roughly the size of our own moon, carrying more liquid water than our entire planet. But the size of the ocean is almost beside the point. What matters is how long it has been there. Earth's oceans are about 3.8 billion years old. Life showed up in them relatively quickly after they formed. Scientists believe Europa's ocean has existed for a similar length of time, possibly longer, kept liquid by a constant source of heat generated by Jupiter's gravity. Here is how that works. Europa orbits Jupiter in a slightly oval path. As it moves closer to Jupiter, the planet's enormous gravity pulls on it harder. As it moves farther away, the pull relaxes. That constant squeezing and releasing flexes the moon's interior the way you might flex a rubber ball. The friction from all that flexing produces heat. Deep inside Europa, rock is being warmed, and that heat rises into the ocean above. This process is called tidal heating, and it has been running continuously since Europa formed. Scientists have no reason to believe it will stop anytime soon. The bottom of that ocean, sitting on rocky seafloor, heated from below by tidal forces, is almost certainly hosting hydrothermal vents. The same conditions that produced the explosion of life around the Galapagos vents in 1977 exist, potentially, on the floor of a moon orbiting Jupiter right now. Scientists cannot confirm the vents are there yet. The ice is too thick and too far away to see through directly, but the chemistry gives strong hints. When Hubble Space Telescope images of Europa were analyzed, scientists spotted what appeared to be plumes of water vapor rising above the surface in several locations. Something from the ocean below was forcing its way up through cracks in the ice. Even more striking were the surface patterns. Europa's ice is covered in long, reddish-brown streaks and cracks that look, at large scale, like the cracked shell of a hard-boiled egg. Those markings come from minerals being pushed up from the ocean below and freezing on the surface. The color suggests sulfur compounds, and sulfur is exactly what hydrothermal vent ecosystems run on. The pressure of the ocean underneath keeps the water liquid despite the deep cold of space above. The salt content helps too. Salt water freezes at a lower temperature than fresh water. Scientists believe Europa's ocean is salty, possibly similar in salt content to Earth's own oceans. All of this means Europa is, by the standards the Mobile Cave and the Galapagos vents established, a plausible home for life. Liquid water, chemical energy from below, a stable environment lasting billions of years. A mission called Europa Clipper launched in 2024 and is now traveling toward Jupiter. When it arrives, it will fly past Europa more than 40 times, scanning the ice, sampling the plumes, and mapping the ocean below. That mission could bring back the first solid evidence of life existing somewhere other than Earth. But Europa is not the most dramatic case in the outer solar system. Another moon is already doing something that Europa only hints at. It is shooting its ocean directly into space. Scientists studying coral reefs on Earth notice something useful. Reefs do not grow at random. They grow where currents bring nutrients, where light reaches through water, and where the chemistry stays in the right range. Reefs are maps of where conditions are good. The underside of Europa's ice shell, they now believe, may work the same way. When warm water rises from the heated ocean floor and hits the cold ice above, it does not just melt straight through, it moves sideways. It carves channels. It hollows out pockets. It creates a chaotic, layered, uneven surface underneath the ice, full of ridges and cavities and sheltered spaces. Scientists modeling this process call it a thermally driven ice architecture. And in those sheltered spaces, chemistry happens. Minerals carried up from the seafloor get deposited in the ice. Hydrogen and oxygen get produced by the interaction of salt water and rock. Simple chemical reactions create compounds that, on Earth, are exactly what microbial life eats. All of this happens at the ice-water boundary, a zone just a few hundred yards thick in places where the temperature, pressure, and chemistry all happen to align. On Earth, the boundaries between two environments, where ocean meets ice, where rock meets water, where light meets dark, are almost always where life concentrates. Evolution favors the edge. If Europa's ice underside works the same way, the most likely place to find life is exactly there, not in the open ocean, not on the floor, but clinging to the ice ceiling in the dark. Europa Clipper is not designed to land. It will not drill through the ice. The ice is too thick, and the engineering challenge is too large for this mission. Instead, it will fly past at low altitude over 40 times, using radar to peer through the ice from above, using spectrometers to analyze whatever is in the plumes, and using cameras to map the surface features in extreme detail. Scientists hope that by mapping the undersurface terrain indirectly, through the patterns visible on top, they can identify the most promising zones for a future lander mission. The areas where ice is youngest, thinnest, or most actively churning are the best candidates. There is a real possibility, seriously discussed among scientists, that the plumes Europa sends up through its cracks carry biological material from the ocean below. Microbes, or pieces of microbes, or the chemical waste products that microbes produce, could be riding up through those cracks and floating above the surface right now. If Europa Clipper flies through one of those plumes at the right time and the instruments pick up the right signals, the mission could detect signs of life without ever touching the ocean itself. That is the plan. That is the hope. But while Europa makes scientists hopeful, another moon in the same solar system makes them almost certain something worth finding is out there. Because that moon is doing what Europa only hints at. It is not hiding its ocean under miles of ice. It is spraying it directly into space, right now, in geysers tall enough to swallow continents. Saturn has over 100 moons. Most are frozen, cratered, and quiet. Enceladus is none of those things. It is a small moon, only about 300 miles across, roughly the size of the state of Arizona. Scientists expected it to be dead, a rock covered in old ice, nothing worth studying closely. Then the Cassini spacecraft arrived at Saturn in 2004 and changed everything scientists thought they knew about that small, quiet moon. Cassini's instruments detected heat coming from the south pole of Enceladus. That was already strange. A moon that small should have lost its internal heat billions of years ago. But the south pole was warm. Then Cassini spotted the plumes. Rising from a series of long cracks near the south pole, called tiger stripes because of their striped appearance from above, were enormous jets of water vapor, ice particles, and gas shooting up into space. Each jet climbed hundreds of miles above the surface. Together, they formed a continuous spray that fed directly into one of Saturn's rings, the E-ring, the faint outermost ring that scientists had never been able to fully explain. Enceladus was building it, particle by particle, every single day. Cassini did not just observe the plumes, it flew through them. In 2008, mission controllers directed the spacecraft into the plume stream, flying just 15 miles above the surface of Enceladus, moving at roughly 18,000 miles per hour, with instruments open and sampling everything in the spray. The data came back over the following months. Scientists went through it carefully, and what they found was not just water. The plumes contained water vapor, as expected. They also contained carbon dioxide, methane, molecular hydrogen, and dozens of organic compounds. Molecules built around carbon, the same basic building block that every living thing on Earth uses. The mix was complex. It was not a simple chemical blowout. It looked like the output of an active chemical system. Scientists identified silica nanoparticles in the spray. Silica is produced when water at very high temperature interacts with rock. On Earth, the only place that process happens at the scale needed to produce those particles is deep inside hydrothermal vent systems. The conclusion was almost unavoidable. Enceladus had an ocean. That ocean sat on a rocky seafloor. Water was circulating through hot rock deep inside the moon, and the products of that circulation were shooting out through the South Pole and into space. Cassini had accidentally found the most accessible ocean in the solar system. Samples were already falling into its instrument bay. The moon was handing them over for free. The only question left was what those samples actually meant for life. And the answer to that question came from one specific ingredient in the plume, one that scientists had not expected, one that, when they found it, made the room go quiet. Molecular hydrogen was the find that changed the conversation. It sounds simple. Hydrogen is the most common element in the universe. Finding it somewhere is not usually news. But finding molecular hydrogen in the plume of Enceladus, in the amounts Cassini measured, meant something very specific. On Earth, molecular hydrogen is produced in large quantities in one particular place. At hydrothermal vents, when superheated water reacts with certain types of rock, the reaction strips hydrogen from the water molecules and releases it as gas. This process is called serpentinization. It runs continuously at every active vent system on Earth, and every living thing near those vents uses that hydrogen as a power source. Microbes at the base of hydrothermal vent food chains pull energy directly out of the reaction between molecular hydrogen and carbon dioxide. The byproduct is methane. This is called methanogenesis. It is one of the oldest forms of life known on Earth, believed to be close to the original life form that appeared billions of years ago. Cassini found molecular hydrogen in the plume. It also found carbon dioxide, and it found methane. All three together, in the right ratios, pointed at one specific explanation. Something inside Enceladus was either running serpentinization reactions on the ocean floor, or something was consuming those ingredients, the same way ancient microbes on Earth do. Scientists could not rule out a purely chemical explanation. Serpentinization alone could produce hydrogen without any life involved. That is the honest answer. But the methane levels were a problem. Purely chemical processes do not typically produce methane in those quantities under those conditions. Biological methanogenesis fits the data better. A team studying the Cassini results published a paper in 2023 stating directly that the methane levels found in the Enceladus plume are hard to explain without biological activity. They were careful. Scientists are always careful with claims like that, but they said it clearly. Enceladus also had salt. The ice particles in the plume contained sodium chloride, ordinary table salt. That meant the ocean was salty, salt water, interacting with hot rock, producing hydrogen, releasing methane, spraying the whole mixture into space. Every ingredient life needs on Earth was present in that spray. Water, chemical energy, carbon compounds, salt, warmth, stability, lasting potentially billions of years. Cassini is gone now. The mission ended in 2017 when scientists deliberately crashed the spacecraft into Saturn to avoid any chance of it contaminating Enceladus with Earth microbes. That choice tells you how seriously scientists took the possibility of life there. They destroyed a perfectly functional spacecraft to protect whatever might be living in that moon's ocean. A follow-up mission is in early planning stages. The idea is to fly through the plume, again with far more sensitive instruments, ones capable of detecting individual biological molecules. If life exists in that ocean, the plume is already delivering the evidence into space. We just need the right tools to read it. And while Enceladus offers a chemical argument for life, the next moon in this story offers something even more unsettling. A world with lakes, rivers, and rain. A world with a complete water cycle. A world where none of those liquids are water at all. Stand on the surface of Titan and look up. The sky is orange, thick, hazy, layered orange, from a smog so dense you cannot see the stars. It is cold enough that the metal parts of your equipment would shatter. The pressure is about 1.5 times what you feel standing at sea level on Earth. But it rains, not water, methane. Liquid methane falls from orange clouds, runs into rivers, fills lakes, and eventually evaporates back into the atmosphere to rain again. Titan has a complete liquid cycle, the only world besides Earth in the solar system that does. The lakes near its north pole are larger than any of the Great Lakes. Some of them may be hundreds of feet deep. The Cassini spacecraft mapped all of this in extraordinary detail before its mission ended. Scientists could trace the riverbeds, measure the lake depths, and watch the weather patterns shift between seasons. Titan is not a barren, frozen rock. It is geologically and chemically active, with complex molecules raining out of the sky, settling on the surface, and possibly reacting in ways scientists are still working to understand. Here is the strange question those lakes raise. On Earth, lakes are where life concentrates. Nutrients, shelter, liquid medium, stable chemistry. Every lake on Earth carries life. So the natural follow-up is, what about Titan's lakes? Methane is not water. Life as we know it uses water as its solvent, the liquid that allows chemistry to happen inside a cell. Water dissolves things easily, moves molecules around, and maintains the right chemical balance for the reactions cells need to run. Methane can theoretically do some of those things too, under the right conditions. It is liquid at Titan's surface temperatures. It carries dissolved chemicals. It allows molecules to mix and interact. Scientists at Cornell University ran a detailed computer model of a theoretical cell membrane that could function in liquid methane. They used chemicals available on Titan's surface. The membrane they designed was stable. It could hold together in liquid methane. It could, in theory, separate an inside from an outside, which is the most basic requirement for a cell to function. They named this theoretical structure an azotosome. It would contain no water and no phospholipids, the molecules Earth cells use for their membranes. It would be built entirely from nitrogen, carbon, and hydrogen. All three are abundant on Titan. The model was just a model. No one has found an azotosome, but the result proved that the chemistry does not break down. A methane-based cell membrane is not physically impossible. What would such a life form eat? Titan's atmosphere constantly produces acetylene and ethane, two carbon-based molecules that rain down onto the surface. A living thing that absorbed those chemicals and combined them with hydrogen from the atmosphere would release energy in the process. That reaction works. Scientists calculated it. No one knows if anything in those methane lakes is actually doing it. A mission called Dragonfly, a rotorcraft designed to fly through Titan's thick atmosphere, is planned for launch later this decade. It will sample the surface in dozens of locations and search for exactly these kinds of chemical signatures. What it finds there could reshape biology as a science permanently. Because if life uses methane instead of water on Titan, it means chemistry has more than one path to producing living things. And the proof that life can bend the rules that radically, it was already sitting in a California lake, waiting for someone to notice. Close your eyes for a second and picture a living cell. Most people picture something like a tiny water balloon, a thin, flexible wall surrounding a watery interior, full of moving parts, chemical reactions firing constantly, energy flowing in and waste flowing out. That picture is accurate for every living thing on Earth. Now, replace the water with methane. Make the temperature drop to -290 degrees Fahrenheit. Remove the oxygen entirely. Keep everything else. Does the chemistry still work? That was the question scientists at Cornell decided to answer seriously in 2015, using computer models to test whether a functional cell structure could operate in liquid methane conditions. The challenge starts with the membrane. Every Earth cell is wrapped in a membrane made of molecules called phospholipids. These molecules have one end that likes water and one end that hates it. In a watery environment, they automatically arrange themselves into a double layer, with the water-hating ends hidden in the middle and the water-loving ends facing out on both sides. That double layer is what forms the cell wall. In liquid methane, water-based membranes fall apart instantly. The chemistry that holds them together requires water to function. So a methane-based cell would need a completely different structure. The Cornell team designed one from scratch, using only the molecules available on Titan. They chose acrylonitrile, a nitrogen-carbon compound found in Titan's atmosphere in significant quantities. When they modeled how acrylonitrile molecules behave in liquid methane, they found that the molecules naturally cluster together into a single-layer membrane. One layer instead of two, but stable, flexible, and capable of forming a closed sphere. That sphere, the azotosome, would be about the same size as a typical Earth cell membrane. From the outside, you might not even notice the difference. The inside would be stranger. No water means no water-based chemistry. The reactions that Earth cells run to produce energy, digest food, and copy themselves all depend on water as the medium. A methane-based life form would need entirely different reaction pathways, ones that run at extremely cold temperatures, in a liquid that dissolves different things than water does. Some of those pathways exist in theory. Acetylene and hydrogen can react to release energy. That reaction is thermodynamically favorable, meaning it releases more energy than it takes in, which is the basic requirement for a fuel source. A cell that could harvest that reaction would have a power supply. Reproducing would be the deepest unknown. Earth cells copy themselves using molecules like deoxyribonucleic acid, which stores information in a specific chemical structure that depends heavily on water to function. What would store information in a methane cell? Scientists genuinely do not know. That is not a gap in the model. That is the frontier. The honest answer is that a methane-based life form remains theoretical. The chemistry allows for it. The conditions on Titan support it. But scientists have not found one. What they have found is something almost as surprising. A living thing on Earth that breaks the rules of biology so thoroughly that for a time, scientists thought it had rewritten them. That discovery happened in a salt lake in California, and the scientific community exploded over it. Before the Cornell model, astrobiologists had a quiet assumption embedded in their work. Life requires a liquid solvent. Water is the best solvent. Therefore, the search for life is the search for water. That chain of reasoning seemed airtight. Water is remarkable. It stays liquid across a wide temperature range. It dissolves an extraordinary variety of chemicals. It participates directly in the reaction cells used to generate energy and build proteins. No other molecule does all of that as well. The assumption was not unreasonable. It was just narrow. The Cornell team was not trying to overthrow biology. They were asking a more specific question. If the chemistry could work in another solvent, what would the cell look like? They picked liquid methane because Titan's surface provides it in enormous quantities, and Titan already sits on scientists' short list of places to study. When the model ran and the results showed a stable membrane, the reaction in the field was immediate. Researchers started revisiting the assumption that water was required rather than preferred. The distinction matters enormously. Required means no water equals no life, end of story. Preferred means life evolved toward water on Earth because water was available, but given a different starting environment, it might have evolved toward something else. Scientists have a name for this distinction now. They call it water chauvinism. The bias toward assuming water is a universal requirement rather than a local solution. Getting past water chauvinism forced a rethink of what the word life actually means. Before the Cornell model, most working definitions of life included something about water-based chemistry. After it, several leading astrobiologists began pushing for a more stripped-down definition. Something like a system that takes in energy, maintains its internal organization, and reproduces with variation. No mention of water, no mention of carbon, no mention of specific chemical reactions. That definition would include an azotosome in methane. It would also include things scientists have not imagined yet. The shift in definition changed how missions get designed. If you search only for water, you build instruments that detect water chemistry. If you search for energy intake and self-organization, you build something different. Instruments that detect unexpected chemical gradients. Instruments that look for patterns that cannot be explained by simple physics. Scientists call these biosignatures, chemical or physical patterns that life creates and that chemistry alone typically does not. On Earth, examples include oxygen in the atmosphere, the specific ratios of carbon isotopes in organic material, and the presence of certain complex molecules in concentrations that random chemistry would not produce. Designing instruments to find those signatures across a range of possible chemistries is the current challenge. It is hard. The search space is enormous. But the Cornell result gave astrobiologists something valuable. It gave them proof of concept. The first door past water-based life opened, and it opened with math that held up to scrutiny. What came next threatened to tear the field apart entirely, because a real discovery, made in a real lake on Earth, appeared to prove that life on our own planet had already crossed into a completely different kind of biology. The scientific community was not ready for it. Mono Lake sits in the California desert, east of Yosemite. It is roughly twice as salty as the ocean and loaded with a chemical called arsenic, which is toxic to almost every living thing on Earth at any meaningful concentration. Scientists had studied organisms surviving in the lake for years. It was unusual but not unprecedented. In November of 2010, a NASA scientist named Felisa Wolf-Simon stood in front of cameras and announced something that sent a shock through the entire scientific world. Her team had taken bacteria from Mono Lake and grown them in a lab environment where arsenic replaced phosphorus. Phosphorus is one of the 6 elements considered absolutely essential to all life on Earth. Every cell uses it. It forms the backbone of deoxyribonucleic acid. Every organism studied before this point required it. Wolf-Simon's team claimed their bacteria had incorporated arsenic into their cellular structure in place of phosphorus. If true, it meant a living thing on Earth had crossed into a completely different biochemistry. Life had broken one of its own fundamental rules, right here at home, in a California lake. NASA called a press conference. Headlines around the world called it the discovery of shadow life, an organism using a fundamentally different set of building blocks than every other known living thing. Then other scientists got hold of the data. The criticism arrived fast and was pointed. Researchers argued that the experimental controls were not tight enough. The bacteria might have been using trace amounts of phosphorus that contaminated the growth medium rather than truly substituting arsenic. The original paper had not ruled that out with enough rigor. Over the next 2 years, multiple independent teams attempted to reproduce the result. None of them found arsenic in the bacterial cell structure. The competing explanation, contamination from trace phosphorus, became the consensus view. The paper was never formally retracted, but the core claim that the bacteria had replaced phosphorus with arsenic was effectively rejected by the field. What the bacteria had actually done was impressive enough on its own. They had survived in arsenic-heavy conditions by using extremely efficient mechanisms to pump the arsenic out of their cells and hold on to every trace of phosphorus they could find. That adaptability alone was remarkable, but it was adaptation, not a new biochemistry. The episode left a complicated legacy. It embarrassed NASA and made scientists more cautious about headline-generating claims in astrobiology. It also demonstrated exactly how the scientific process is supposed to work. A claim is made, other teams test it, the community corrects itself, and it raised a question that nobody dropped. If life could almost rewrite its own chemistry, even if this particular case turned out not to be that, then somewhere in the universe, had anything actually done it? Had any organism ever replaced phosphorus, or carbon, or water with something else and survived? Scientists still do not know. But the search for an organism that truly breaks the rules did not end with Mono Lake. It continued in a very different direction. Scientists stopped looking for chemistry and started looking for survival, and the organism that won that competition was barely visible to the naked eye and had already survived conditions that should have made life impossible by any standard. Pick up a handful of moss from a garden wall. Inside it, invisible without a microscope, are tardigrades. Hundreds of them, possibly thousands, moving slowly through the water film between the moss fibers, eating algae, reproducing, living out lives of extraordinary biological toughness in a world most of us never think about. Tardigrades are tiny, usually around a 20th of an inch long. They have 8 stubby legs and a round, segmented body that looks like a miniature water bear, which is actually one of their common names. They have a mouth designed to pierce plant cells and suck out the contents. They have existed for over 500 million years. They outlived the dinosaurs, multiple ice ages, and every mass extinction event in Earth's history. They do this by cheating death at the cellular level. When conditions turn lethal, a tardigrade can enter a state called cryptobiosis. It pulls all the water out of its body, retracted into a dry husk about a third of its normal size, and halts almost all biological activity. Metabolism drops to less than 1/10,000th of its normal rate. The tardigrade is alive, technically, but barely doing anything at all. In that state, it has survived temperatures ranging from -320 degrees Fahrenheit to 300 degrees Fahrenheit, a range that covers conditions from the surface of Pluto to a kitchen oven. It has survived pressure 6 times greater than the deepest part of the ocean floor. It has survived doses of radiation roughly 1,000 times higher than what would kill a human adult. Scientists have revived tardigrades after they spent 30 years frozen in Antarctic ice. They have revived them after they were dehydrated for a decade. One team revived tardigrades from a museum specimen that had been sitting dry in a drawer for over 100 years. The cellular mechanism behind this is still not fully understood. When a tardigrade enters cryptobiosis, it produces specific proteins that replace the water in its cells with a glassy solid substance that essentially freezes the cell's internal structures in place, preventing damage until water returns. No other animal on Earth uses this specific biological trick. Scientists have found tardigrades in the Himalayan mountains at altitudes above 18,000 feet, in hot springs, in Antarctic dry valleys, in the deep sea sediment, in tropical rainforests, and on roof tiles in cities. Every continent, including Antarctica, every major ecosystem. The one environment scientists had not tested was space itself. That changed in 2007 when a team launched tardigrades on a spacecraft and deliberately exposed them to open space for 10 days. The temperature swings, the hard vacuum, the ultraviolet radiation, the cosmic radiation, full exposure, no protection. When the spacecraft returned, a portion of the tardigrades were alive. That result was stunning, and it immediately opened a question that scientists had been arguing about for years, a question about whether life on Earth might have come from somewhere else entirely. The experiment was called TARDIS, short for Tardigrades in Space. It launched as part of a European Space Agency mission in 2007. The tardigrades were loaded into small containers mounted on the outside of the spacecraft, completely exposed to the space environment. No shielding, no atmospheric protection. Direct contact with the vacuum, the radiation, and the temperature extremes of low Earth orbit. 2 groups of tardigrades went up. One group was exposed to the vacuum and cosmic radiation, but protected from the most intense ultraviolet rays from the sunday. The other group received the full treatment: vacuum, cosmic radiation, and direct solar ultraviolet exposure. After 10 days, the spacecraft returned to Earth. Scientists examined the tardigrades. The first group, protected from the most intense ultraviolet, had a survival rate that stunned the team. A significant portion had survived, and several of those survivors went on to reproduce successfully after returning to Earth. Their eggs hatched, the offspring were healthy, the animals had endured space and come back to live normal lives. The second group, hit with full ultraviolet exposure, fared worse. Most did not survive. The ultraviolet radiation damaged their deoxyribonucleic acid beyond what even cryptobiosis could protect against. But the result from the first group mattered enormously. It proved that a living animal could survive open space for at least 10 days if shielded from the most intense radiation. And inside a rock, even a thin rock, there is natural shielding. Ultraviolet radiation cannot penetrate stone. This is where the debate about planetary travel opens up. When large objects hit planets, the impact throws material into space at tremendous speed. Rocks, dirt, and anything inside them can escape a planet's gravity entirely and travel through space for thousands or millions of years before landing somewhere else. Scientists have confirmed this happens. Meteorites found on Earth have been traced back to Mars. One found in Antarctica in 1984 contains rock that solidified on Mars roughly 4 billion years ago. It traveled through space and landed in Antarctica, where humans found it in 1984. The question the tardigrade experiment raised is this: could a microorganism survive that journey? Locked inside a rock, shielded from ultraviolet radiation, in a cryptobiosis-like state for millions of years of travel, and then survive the heat and impact of landing on another planet? The tardigrade data says maybe. Scientists have also exposed bacterial spores to simulated Mars transfer conditions in labs. Some survived. The idea has a name. It is called panspermia, and it is older than most people realize. And one specific piece of evidence, a rock from Mars found in a frozen field in Antarctica, launched the idea from interesting hypothesis into full scientific firestorm. What scientists found inside that rock, and what happened at the NASA press conference where they announced it, is one of the most dramatic stories in the entire history of space science. The idea sounds like science fiction. Life hitching a ride on a rock, flying through the void between planets, surviving impact, landing somewhere new, and starting over. But the math holds up. Panspermia, the transfer of life between planets or star systems via natural debris, has been studied seriously since the 19th century. The Swedish chemist Svante Arrhenius proposed in 1903 that microscopic spores could be pushed through space by the pressure of starlight itself. Radiation pushes. It is not dramatic, but it is real. Scientists confirmed that light exerts a small but measurable force on particles. The problem with Arrhenius's version was radiation damage. Spores traveling through open space would be exposed to ultraviolet and cosmic radiation for thousands of years. Deoxyribonucleic acid would degrade. Nothing would survive. More recent versions of the idea account for that. The lithopanspermia model proposes that life travels inside rocks, which provide shielding. When a large asteroid or comet hits a planet, the impact ejects material at speeds that can exceed the planet's escape velocity. Some of that material ends up in space. Some of it eventually lands on other planets. Scientists calculated how often this happens between Earth and Mars. The result was surprising. Given the frequency of large impacts over billions of years, tens of thousands of rocks may have transferred between the two planets in each direction. Earth rocks have hit Mars. Mars rocks have hit Earth. The journey time matters. A rock ejected from Mars can reach Earth in as little as a few months if the orbital geometry is right, or as long as tens of millions of years if it takes a longer path. For bacterial spores in the right state of dormancy, a few months of travel inside a shielded rock is clearly survivable. Scientists have revived spores sealed in amber for 40 million years. The harder question is star-to-star transfer. Within a solar system, the distances are manageable. Between solar systems, rocks would travel through interstellar space for millions to billions of years, with no planetary gravity to eventually catch them. Radiation exposure over that timescale would likely destroy most biological material. However, scientists have calculated that during the early period of our solar system, the Sun was much closer to its neighboring stars. The whole neighborhood was packed tighter. Transfer times between solar systems 4 billion years ago may have been far shorter than they would be today. If life appeared early in one system, it had a window to spread. Some scientists have even proposed that life did not originate on Earth at all, that Earth received the starting material from Mars or from somewhere farther out, and that everything alive on this planet is technically the descendant of a visitor. That idea is unresolved. Scientists are honest about that. Finding life on Mars that shared a common ancestor with Earth life would support it. Finding life on Mars with completely independent chemistry would mean life appeared independently twice in the same solar system, which would be equally extraordinary. One rock found in Antarctica may already carry the answer. A geologist named Roberta Score was working in the Allan Hills region of Antarctica in December of 1984, collecting meteorites for a survey team. The ice fields there are excellent hunting ground. Meteorites that land in Antarctica are preserved by the cold and then carried slowly toward the edge of the ice sheet by glacial movement, concentrating in predictable locations. Score picked up a potato-sized rock. It was heavier than it looked. The outside was coated in a dark, glassy layer that forms when a rock passes through an atmosphere at high speed. She bagged it and labeled it for later analysis. The rock sat in a freezer at the Johnson Space Center for years. When scientists finally analyzed it in depth in the 1990s, they traced its origin to Mars. The ratios of gases trapped inside matched the Martian atmosphere as measured by Viking landers. The rock had formed on Mars about 4 billion years ago, been blasted off the surface by a large impact, traveled through space, and landed in Antarctica roughly 13,000 years ago. It had been sitting in the ice since then. Inside the rock, scientists found several things that together made an extraordinary pattern. There were microscopic worm-shaped structures, roughly 100 times smaller than a human hair in diameter, that resembled the shapes of fossilized bacteria found in ancient Earth rocks. There were carbonate globules, round chemical deposits of the type that form in liquid water. There were specific organic compounds called polycyclic aromatic hydrocarbons, which on Earth are associated with the decay of biological material. And there were tiny crystals of magnetite, a mineral that certain bacteria on Earth produce inside their own cells to sense magnetic fields. Each piece of evidence, considered separately, had a non-biological explanation. Unusual rock chemistry could produce worm-shaped structures. Organic compounds form through purely chemical processes in space all the time. Magnetite crystals appear without biology. Together, the pattern was harder to dismiss. In August of 1996, NASA Administrator Dan Golden and a team of scientists stood before cameras and announced that they might have found evidence of ancient life on Mars. Not a certainty, a possibility, but a real one, supported by multiple independent lines of evidence in the same rock. The room erupted. Newspapers ran the story on front pages worldwide. The White House issued a statement. The scientific community started arguing immediately. Over the following decade, researchers proposed non-biological explanations for every element of the evidence. The worm shapes were too small to contain the cellular machinery a bacterium requires. The organic compounds might have entered the rock after it landed in Antarctica. The magnetite crystals had features that differed from the biologically produced versions. The consensus shifted. Most scientists today do not believe the rock proves life once existed on Mars. But they do not all agree it proves the opposite either. A small number of researchers still argue that the totality of the evidence, all of it together in one rock, is harder to explain without biology than with it. The rock sits in a freezer. The argument continues, and the question it raised, whether Mars once had conditions where life could have started, remains one of the most actively studied problems in planetary science. What came next in the search for life moved away from rocks and into radio waves, and the signal that arrived in 1977 still has no accepted explanation. On the night of August 15th, 1977, a radio telescope in Ohio called the Big Ear was scanning the sky on autopilot. Nobody was watching in real time. The telescope printed its data onto long sheets of paper, and researchers would review those printouts days later, looking for anything unusual in the streams of numbers. Astronomer Jerry Eman was going through one of those printouts about a week after the scan. The data was mostly noise, the kind of low-level radio background the universe constantly produces. Then he saw a column of numbers that made him stop. The signal was strong, dramatically stronger than the background noise surrounding it. It climbed to the maximum value the telescope could register, held there, then faded back down over the course of 72 seconds. exactly the amount of time the telescope's beam would take to sweep across a fixed point in the sky as Earth rotated. Emmons circled the numbers in red pen and wrote a single word in the margin. Wow. The signal had characteristics that scientists searching for alien transmissions specifically hoped to find. It came from a region of the sky near a star similar to our Sunday. It arrived at a frequency very close to one that scientists had long theorized an advanced civilization might use for communication. The frequency at which hydrogen, the most common element in the universe, naturally emits radiation. Any civilization that understood basic physics would know that frequency. Choosing it for a signal would be a logical beacon. The signal showed no characteristics of terrestrial interference. It was narrow in frequency, which natural cosmic sources rarely are. It did not repeat during the 72 seconds, suggesting it was steady rather than pulsed. And it came from deep space, not from Earth or any known spacecraft. Big Ear pointed back at the same region of sky dozens of times over the following weeks and months. The signal never returned. Scientists have proposed explanations. A comet releasing hydrogen gas was suggested in 2017 as a possible natural source. The team behind that proposal pointed to two comets that were in roughly the right area of sky at the time. Critics pointed out that the frequency match was imperfect and the brightness was too high for that explanation to work cleanly. No explanation has achieved consensus. In 2012, a group of scientists sent a response toward the coordinates the signal came from. It contained greetings in 100 different languages, music, and mathematical information. If the signal came from a civilization roughly 175 light-years away, the response will arrive there around 2189. Nobody expects a reply in our lifetimes. The WOW signal sits in the history of science as the most compelling unexplained candidate for extraterrestrial contact ever recorded. A single burst, 72 seconds, a red pen circle in a margin, and decades of silence after. But what if the problem is not that no one is talking? What if the problem is that we have been listening on the wrong channels the whole time? The search for extraterrestrial intelligence has been scanning radio frequencies since 1960. That is over 60 years of listening, thousands of hours of telescope time, millions of dollars in equipment and analysis, and the result, in terms of confirmed signals, is zero. Some people hear that and conclude the universe is empty. That conclusion misses something important. The radio frequency range that scientists chose to search was not random. It was built around what humans happen to use. Radio waves travel fast, cover enormous distances, and pass through gas and dust without much interference. We use them for television, for satellites, for communications of every kind. So when scientists first designed the search for alien signals, they assumed any advanced civilization would also discover radio and use it similarly. That assumption is already looking shaky on our own planet. Human civilization shifted away from strong radio broadcasts within decades of inventing them. Analog television signals, the kind that blasted strongly into space from the 1950s onward, are being replaced by compressed digital signals that are far quieter. Our radio bubble, the sphere of detectable radio noise expanding outward from Earth at the speed of light, is actually dimming as our technology changes. An alien civilization that went through the same progression might have broadcast strongly for a century or two, then gone quiet. From a distance, they would look like a faint blip followed by silence. We might have already missed that window. The search space problem is enormous in another way. The galaxy contains an estimated 400 billion stars. The radio frequency range covers an almost infinite number of possible channels. Scientists have scanned a small fraction of both. The astronomer Jill Tarter described the situation this way: searching for alien signals so far is like filling a bathtub with ocean water and concluding the ocean has no fish because nothing swam into that bathtub. Beyond radio, scientists increasingly argue that advanced civilizations might communicate through methods our technology cannot yet detect. Neutrinos, particles that pass through entire planets without stopping, could carry signals. Gravitational waves, Ripples in space itself could be modulated. Laser pulses in specific wavelengths could be aimed between star systems with pinpoint precision and be completely invisible to a radio telescope. Some researchers have proposed that a civilization advanced enough would communicate in ways so compressed and efficient that the signal would look like random noise to any receiver not specifically designed to decode it. To us, it would be indistinguishable from background static. The honest answer is that 60 years of radio silence tells us remarkably little about whether life exists out there. It tells us something about what we chose to listen for and how long we looked. It does not tell us about the full range of what might be speaking. But the silence does raise one question that scientists have not been able to shake for decades. A question that starts with a simple observation. The universe is 400 billion stars in just our galaxy, billions of galaxies beyond that, billions of years of time for life to develop. So where is everyone? In 1950, the physicist Enrico Fermi was eating lunch with colleagues at Los Alamos when the conversation turned to extraterrestrial life. Fermi listened for a moment, did some mental calculations, and asked a question that has driven scientists and philosophers mad ever since. Where are they? The universe is roughly 13.8 billion years old. Our sun formed about 4.6 billion years ago. That means billions of years passed before our solar system even existed, during which other stars formed, other planets developed, and other civilizations potentially rose. Given those timescales, even a civilization that travels at 1% of the speed of light could colonize every star in the Milky Way galaxy in about 10 million years. 10 million years is nothing on cosmic timescales. Humanity has existed for less than half of that. So if even one civilization arose and expanded outward, we should see signs of them everywhere. We should have been visited. We should be picking up their signals. The galaxy should be obviously full of life. It appears empty. Scientists call this the Fermi paradox, and over the decades they have developed serious answers to it. The first answer is that we are rare. Complex life requires such an improbable set of conditions: the right star, the right planet, the right moon, the right chemistry at the right time, that it may have appeared only once or a handful of times in the observable universe. We are here, and we are alone. The second answer is the great filter. Some step in the development from simple chemistry to spacefaring civilization is almost impossibly hard to cross. Every civilization that reaches a certain point gets wiped out, either by their own technology, by a natural catastrophe, or by something we cannot imagine yet. The third answer is that they are here, but we cannot recognize them. A civilization a billion years older than us might operate on scales and in ways that are invisible to our current instruments, the way an ant cannot perceive a highway. The fourth answer is that the universe is young. We may be among the first complex civilizations to arise, early arrivals in a universe that will be producing intelligent life for trillions of years to come. The fifth answer is that space is too large. Even if civilizations are common, the distances between them are so vast that contact is essentially impossible. Every civilization develops and dies in isolation, never knowing the others existed. The sixth answer is that they are hiding. A civilization that has been watching the universe long enough knows that announcing your presence is dangerous, so everyone stays quiet. The seventh answer is that they are already here, embedded in our data, our communications, or our planet in a form we have not yet identified. The eighth answer is that we are looking for the wrong things entirely, and life is everywhere around us in forms we have not yet learned to recognize. None of these answers is proven. Every one of them has serious scientists who hold it. The sixth answer, that everyone is hiding on purpose, is the most disturbing, because if it is true, there is a reason for the hiding, and that reason involves exactly the kind of universe that none of us want to live in. The theory comes from a thought experiment. Imagine you are alone in a dark forest at night. You cannot see anyone else, but you know others are in the forest. You do not know who they are, what they want, or how dangerous they are. You have limited resources, You cannot communicate safely. What do? You stay quiet. You stay hidden. You eliminate any threat before it can eliminate you. Chinese science fiction author Liu Cixin built this into a novel, but the underlying logic is also discussed seriously in scientific circles as a possible resolution to the Fermi paradox. Several physicists and astrobiologists have engaged with it as a genuine hypothesis rather than just a story. The reasoning goes like this. Any civilization advanced enough to travel between stars or broadcast across the galaxy has also developed weapons of almost unimaginable destructive power. Resources in the universe are finite. Two expanding civilizations will eventually compete. The civilization that strikes first has an enormous survival advantage. So, every civilization that understands this dynamic will choose to hide its location and destroy any other civilization it detects before that civilization can detect and destroy it first. The logic is cold, but it holds together. If this is true, sending the WOW signal response in 2012 was potentially a serious mistake. Pointing radar at nearby star systems, which scientists have done multiple times in the past 50 years, was potentially a serious mistake. The moment any civilization detects our signals, the clock starts. Some scientists have argued against deliberately broadcasting towards space for exactly this reason. Stephen Hawking publicly warned against it multiple times before his death. He compared humanity reaching out to alien civilizations to Native Americans greeting Columbus, a meeting that did not go well for the less technologically advanced party. Others argue the concern is overblown. Any civilization capable of crossing interstellar distances would need resources so large that destroying Earth would cost more than it gains. Predatory expansion at that scale stops making sense when you can harvest uninhabited systems far more efficiently. The debate is unresolved, but it has a practical consequence. If the dark forest theory is even partially right, the silence we observe is the sound of everyone hiding, and the first civilization to break cover pays for it. There is a related theory that sits one step further along the same path of reasoning, one that suggests civilizations do not just hide, they get eliminated, and that the evidence for this might already be written into the history of life on Earth. It is called the Great Filter. And the question it raises is whether we have already crossed the hardest obstacle to survive or whether it is still ahead of us. Robin Hanson, an economist and researcher, published the Great Filter Theory in 1998. The core idea is simple. Something filters civilizations out of existence. The universe should be full of them, and it appears empty, so something is killing them off before they become visible or before they can spread. The critical question is, where in the timeline the filter sits? Imagine the development of a spacefaring civilization as a series of steps. Simple chemistry forms. Single-celled life appears. Complex cells with a nucleus develop. Multicellular organisms arise. Sexual reproduction begins. Complex animals evolve. Intelligent tool-using creatures emerge. Industrial civilization develops. The civilization survives its own technology long enough to spread to other star systems. Each step is unlikely. Each step depends on the one before. If any single step is nearly impossible to complete, that step is the Great Filter. Almost nothing gets through it. Here is the unsettling split. If the Great Filter is behind us, we are safe. Humanity already crossed the nearly impossible step. Maybe it was the formation of the first cell. Maybe it was the development of the complex nucleus that makes up every animal cell on Earth, an event that may have happened only once in Earth's history. If that step is the filter, it means simple life might be scattered across the universe, but complex life is extraordinarily rare. We won the lottery. If the Great Filter is ahead of us, we are in serious danger. Civilizations routinely reach our stage of development and then fail to survive. They destroy themselves with their own technology. Climate change, engineered pandemics, nuclear war, artificial intelligence gone wrong. Something kills every civilization that reaches a roughly modern level before it can escape its home planet. Finding simple microbial life on Mars or Europa would be terrible news under this framework, because if simple life is common, the filter is not in the early steps. It sits somewhere later in the chain, and every civilization that got where we are eventually hit it. The discovery of complex life elsewhere would be even more terrifying. It would mean the filter sits at a step that complex animals routinely cross, which means it is very close to us in the sequence, which means we are almost certainly about to hit it. Some researchers argue that the Great Filter is already visible in current events. Humanity has had nuclear weapons capable of ending civilization since the 1960s. Engineered biology is advancing toward capabilities that small groups could eventually misuse. Artificial intelligence is developing faster than governance can track. Climate change is straining the systems that support large populations. Every one of those is a candidate filter, not a certainty. A candidate. The best outcome, the one scientists searching for alien life quietly hope for, even though it contradicts their professional goals, is to find absolutely nothing. No microbes on Mars, no chemistry in the European Ocean, no biosignatures on any planet around any star. A universe that appears, by every measure, empty of other life, because that emptiness would suggest we already passed the hard part. What we choose to do next with that possibility, and with the technology to search or to broadcast or to build, may determine which side of the filter we end up on. Meanwhile, scientists are no longer limited to listening for radio signals. They are hunting for something far more dramatic. The physical marks a truly advanced civilization would leave on the universe itself. A civilization 1,000 years ahead of us would look, from our current vantage point, essentially magical. We have had electricity for roughly 150 years, radio for just over 100 years, space travel for less than 70 years. In 1,000 years, assuming we survive, we will likely be doing things that a person from the year 2000 could not conceptualize. Now imagine a civilization 1 million years ahead of us, or 1 billion. The question scientists are now seriously asking is, what marks does a civilization that old leave on the universe around it? Those marks are called technosignatures, and hunting for them has become a growing field within astronomy. The most obvious technosignature is light pollution. Cities on Earth are visible from orbit at night. A planet densely covered in artificial light would show a different signature from space than a natural planet. Scientists have modeled what this would look like in the spectral data a distant telescope collects. Artificial light sources have different wavelength profiles than reflected starlight or volcanic activity. Heat is another one. Every energy conversion produces waste heat. A civilization using enormous amounts of energy would radiate that heat, and it would be detectable as an infrared excess around their planet or star system. An advanced enough civilization might actually alter the overall temperature of their home planet in measurable ways. Atmospheric pollution is a third. Certain industrial chemicals, ones that have no known natural source, would appear in a planet's atmosphere if industry was producing them. Some researchers have suggested looking for nitrogen dioxide specifically, a byproduct of combustion that exists in Earth's atmosphere, primarily due to industrial activity and traffic. Scientists have also proposed looking for the chemical signatures of large-scale mining or terraforming. Moving enough rock to change a planet's surface composition would leave traces detectable in light reflected from that planet. All of these technosignatures are passive. They do not require any civilization to be trying to communicate. They are the exhaust of existence, the marks that large-scale activity leaves whether anyone intends it or not. The James Webb Space Telescope, now operational, can analyze the atmospheric chemistry of planets orbiting distant stars. It was designed primarily to look for biosignatures, the chemical marks of biology. But the same instruments can look for technosignatures. The data comes back either way. Some of the most promising proposals involve looking for signatures that are clearly artificial in pattern, mathematical sequences, structures with geometric regularity, or energy emissions that follow patterns no natural process would produce. One proposed technosignature sits at the absolute extreme of what a civilization could build. A structure so large, it would be visible across interstellar distances, wrapped around a star, harvesting its entire energy output. Scientists have already found candidate stars that behave in ways no known natural process fully explains. Freeman Dyson was a physicist who in 1960 published a paper with a simple but staggering idea. Any civilization that survives long enough will eventually need more energy than a single planet can provide. Stars produce an almost incomprehensible amount of energy, most of which streams off in all directions and is wasted in empty space. A sufficiently advanced civilization would eventually build structures to capture that energy, wrapping around the star in some configuration to collect as much of its output as possible. Dyson proposed looking for the heat signature such a structure would produce. A star surrounded by a massive shell of material would absorb the star's visible light and re-emit it as infrared radiation, heat. From a distance, such a star would look dim in visible light, but extremely bright in infrared, an unusual combination that natural processes do not typically produce. Astronomers call these hypothetical structures Dyson spheres. And over the decades, teams have periodically searched for their signatures in stellar data. In 2015, a star called KIC 8462852, nicknamed Tabby's star, after astronomer Tabetha Boyajian, who led the team studying it, made international headlines. It was dimming in patterns that no known natural explanation fully accounted for. The dimming was irregular, non-repetitive, and reached as deep as 22% of the star's total light, far more than any orbiting planet would cause. Alien megastructures became the most discussed explanation in popular science for months. Scientists kept studying Tabby's star. Eventually, detailed color measurements of the dimming showed that shorter wavelengths of light were blocked more than longer wavelengths. Solid objects, like a megastructure, would block all wavelengths equally. The wavelength dependence pointed instead to clouds of fine dust, possibly from comets or from some form of planetary breakup. The megastructure idea faded. But Tabby's star is not the only candidate. Two other stars in astronomical datasets have been flagged for producing infrared excess in patterns that do not match standard stellar models. Neither has been explained definitively. Both are being watched. A systematic search through data from the Wide Field Infrared Survey Explorer, a space telescope that surveyed the entire sky in infrared, found several hundred stars with unusual infrared excess worth follow-up study. Most will have mundane explanations: disks of dust, nearby galaxies coincidentally aligned, instrument artifacts. A small number have held up to follow-up examination without resolution. Scientists are clear that no Dyson sphere has been confirmed. The bar for that claim is extraordinarily high, but the search is no longer considered fringe science. Several major research universities now have active programs searching for large-scale astro-engineering signatures in archival telescope data. The idea of a civilization advanced enough to engineer stars is almost impossible to fully absorb. But what happened on a planet much closer to home, a planet we can see with the naked eye, is just as hard to explain. In 2020, scientists detected something in the atmosphere of our nearest planetary neighbor that had no business being there. Jane Greaves was not looking for signs of life when she pointed the James Clerk Maxwell telescope at Venus in 2017. She was testing a new instrument. Venus was a convenient, bright target. She planned to use the observation as a calibration check and move on. Instead, she found something in the spectral data that stopped her for months. Phosphine, a molecule made of one phosphorus atom and 3 hydrogen atoms. It smells like garlic and rotting fish and is toxic enough that militaries once considered it as a chemical weapon. It is also the molecule that bacteria in oxygen-free environments on Earth produce as a byproduct of their metabolism. In the clouds of Venus, floating at an altitude of about 37 miles, where the temperature and pressure are surprisingly close to Earth's surface conditions, the instruments detected phosphine at concentrations of about 20 parts per billion. 20 parts per billion sounds small. It is not small for phosphine. On Earth, Phosphine in the atmosphere occurs in tiny amounts in certain environments, primarily above swamps and landfills where anaerobic bacteria produce it. Even in those environments, the concentrations are far lower than what the Venus data showed. Greaves brought in a team. They checked the data repeatedly. They ruled out instrument error. They analyzed every known chemical process that might produce phosphine in Venus's atmosphere. Lightning strikes could produce trace amounts. Volcanic activity could produce trace amounts. Meteorite impacts could produce trace amounts. None of those processes came close to matching the concentrations in the data. In September of 2020, the team published the finding and held a press conference. The announcement landed like a shockwave. Within days, scientists from around the world began picking the paper apart. A team analyzing a different telescope's data of Venus initially claimed they found even higher concentrations. Then, that same team revised their findings downward. The original team re-examined their data and found a calibration error that reduced their phosphine estimate. The revised number was still significant but lower than the original headline figure. The argument about whether the phosphine is actually there and in what quantities is still active. Multiple telescope teams are currently observing Venus to get a cleaner measurement. The deeper argument about what could produce it, if it is there, is also still active. Some researchers have proposed exotic atmospheric chemistry that might manufacture phosphine without biology. So far, none of those proposals have fully accounted for the signal. The most radical version of the hypothesis involves something unexpected about Venus's history. Because if Venus once had conditions where life could start, then whatever is producing phosphine in those clouds today might be something that survived a catastrophe far worse than anything life on Earth has ever faced. 4 billion years ago, Venus may have looked a lot like Earth. Scientists running climate models of early Venus found that the planet likely had liquid water on its surface for somewhere between 1 and 3 billion years. The sun was dimmer then. Venus rotated much more slowly than Earth, which would have helped distribute heat. The conditions, while harsh by Earth standards, may have been compatible with life. Then something changed. Scientists call it a runaway greenhouse event. Volcanic activity on a massive scale pumped carbon dioxide into the atmosphere. Carbon dioxide traps heat. The atmosphere grew hotter. Hotter temperatures caused the oceans to evaporate faster. More water vapor in the atmosphere trapped even more heat. The cycle accelerated. The oceans boiled. The water vapor itself eventually broke down in the upper atmosphere and escaped into space. Venus today has a surface temperature of about 900 degrees Fahrenheit and an atmospheric pressure 90 times higher than Earth's. The surface would melt lead. No liquid water survives. But the clouds are different. At roughly 30 to 40 miles altitude, the temperature in Venus's cloud layer drops to between 30 and 160 degrees Fahrenheit. The pressure is close to what you feel standing at sea level on Earth. Sunlight reaches the clouds directly. If you were floating there, without any protective equipment, the temperature would be manageable. The problem is the clouds themselves. They are made of concentrated sulfuric acid droplets. Sulfuric acid destroys most organic molecules on contact. Any life floating in those clouds would need to survive in an environment of extremely strong acid. Some bacteria on Earth tolerate high acid conditions. None tolerate pure, concentrated sulfuric acid. But 1 billion years of evolution under different pressures might produce something we have not seen. A group of researchers at MIT and Cambridge proposed in 2020 that microbial life could potentially survive in the Venusian cloud layer if it evolved the right chemical defenses. The sulfuric acid would need to be kept outside the cell, which would require a specialized membrane. The life form would need to harvest energy from sunlight or from the chemical reactions in the clouds. And it would need to survive the cloud droplets evaporating and reforming constantly as they cycle through different altitudes. The researchers also proposed a survival mechanism. When a cloud droplet falls below the habitable altitude and starts to dry out, the microbe inside it could enter a dormant spore state and wait to be lofted back up to the habitable zone by the atmosphere's circulation patterns. This cycle of dormancy and activity, driven by the vertical circulation of the Venusian atmosphere, could sustain a population indefinitely. Scientists are careful to describe this as a hypothesis supported by plausible chemistry. No organism has been found in the clouds of Venus, but the phosphine signal, if confirmed, would be consistent with exactly these types of organisms, producing it as a metabolic byproduct. And if life survives in the clouds of Venus, it is not the only place in the solar system where floating aerial life has been seriously proposed. The cloud layer of Venus is the most Earth-like environment outside of Earth in the inner solar system. That sentence surprises most people, because Venus is usually described as our worst-case scenario, a hellish, scorching, acid-soaked nightmare. On the surface, that is accurate, but altitude changes everything. At 37 miles up, inside the dense cloud deck, the temperature hovers around 160 degrees Fahrenheit at the lower edge and drops to freezing temperatures at the upper edge. Pressure is close to 1 atmosphere. Sunlight is abundant. Carbon dioxide, water vapor, sulfur dioxide, and other chemicals are all available in significant quantities. Every ingredient a photosynthesizing microbe might need is present if you can solve the acid problem. Researchers at MIT modeled the chemistry in detail. They found that certain chemical reactions available in the cloud environment could supply energy to a microbe without requiring the microbe to directly contact or ingest sulfuric acid. The reactions involve sulfur compounds cycling between different oxidation states, a process driven by the energy of sunlight hitting the cloud layer. The modeled energy yield was comparable to what photosynthetic bacteria extract from sunlight on Earth. A cloud-dwelling organism could, in theory, run its metabolism entirely on those reactions. The water question is more challenging. Sulfuric acid is extremely good at pulling water molecules out of biological structures. A microbe in the Venusian clouds would be under constant chemical pressure to lose its internal water to the surrounding acid. The proposed solution involves a protective layer of some kind, a coating or modified membrane that sulfuric acid cannot penetrate efficiently. Such coatings exist in extreme acid-tolerant bacteria on Earth, though none have been tested against the concentrations found in Venus's clouds. What makes the aerial life hypothesis compelling beyond just the chemistry is the phosphine signal. Phosphine has no accepted non-biological source in the quantities detected. If those quantities are confirmed by further observation, aerial microbes metabolizing sulfur compounds and releasing phosphine as a byproduct would fit the data. A series of proposed missions to Venus are now moving through planning stages at various space agencies. One concept sends an atmospheric probe that floats through the cloud layer for weeks or months, sampling the chemistry directly. Another proposes a sample return mission that captures cloud material and brings it back to Earth for analysis. If those missions find complex organic molecules in the cloud droplets, or isotope ratios that only biology produces, or cellular structures of any kind, the implications extend far beyond Venus. Because a second independent origin of life in our own solar system, even in a cloud layer, would statistically transform the probability of life across the rest of the universe. If it happened twice here, in one system, in two very different environments, the number of inhabited worlds in the galaxy explodes. But the solar system still holds one more category of possible inhabited world that most people have never considered. Worlds that belong to no star at all. Most planets orbit stars. That is the picture most people carry. A sun, planets circling it, the whole system moving together through the galaxy. That picture is incomplete. Astronomers have discovered that a significant number of planets have been ejected from their home solar systems and now drift alone through interstellar space, with no star to orbit and no sunlight to warm them. Scientists call them rogue planets. Some estimates suggest there may be billions of them in the Milky Way alone, outnumbering the planets that orbit stars. On the surface, a rogue planet sounds like the most hostile possible environment for life. No sunlight means no energy from above. No star means no magnetic field protection from cosmic radiation. Temperatures on the surface would plummet toward absolute zero over billions of years. But the surface is not where the interesting activity happens. A rogue planet large enough, roughly similar in size to Earth, generates its own internal heat through radioactive decay in its core. That heat is not enough to warm a surface exposed to the cold of deep space. But underneath a thick enough atmosphere, or thick enough ice shell, it could maintain liquid water indefinitely. The key is the atmosphere. Scientists modeling rogue planet conditions found that a planet retaining a thick, hydrogen-rich atmosphere, the kind that forms during early planetary development, could use that atmosphere as an insulating blanket. Hydrogen is extremely effective at trapping heat. A hydrogen atmosphere 100 times thicker than Earth's current atmosphere could theoretically keep surface temperatures above freezing even without any star at all. Radioactive decay in the planet's interior would provide a steady heat source. The same tidal flexing that warms Europa might not apply, since there is no nearby massive planet to cause tidal forces. But radioactive decay alone, running for billions of years, can maintain a warm interior. Liquid water could persist in underground oceans, heated from below, covered by a thick ice shell and insulated from the cold above by a hydrogen atmosphere. The same basic setup as Europa, minus the tidal heating, plus a thick blanket of gas. Hydrothermal vents could operate on the seafloor of that underground ocean, driven by the planet's own internal heat. The same chemistry, the same energy source, the same potential for the same kind of microbial life that scientists found around the Galapagos vents in 1977. A rogue planet with this setup could carry life indefinitely, drifting through the cold dark between stars, completely undetectable by telescopes that look for planets by searching for the dimming they cause when they pass in front of a star. Finding rogue planets is extremely difficult. Detecting life on them is currently beyond our capabilities. But the universe has had over 13 billion years to scatter them across enormous distances, and if any of them carry life, that life has been sealed away from the rest of the cosmos, evolving in total isolation for timescales that make Earth's history look brief. And that isolation opens the deepest question in this entire story. Not just where life might exist right now, but how much of cosmic history we have already missed. 10 billion years. That is the gap between the beginning of the universe and the moment the first living thing appeared on Earth. 10 billion years during which stars formed, burned through their fuel, exploded, scattered heavy elements across space, and gave those elements time to assemble into new planetary systems. Earth got a late start. The first generation of stars in the universe contained almost no heavy elements. The periodic table was mostly empty: hydrogen, helium, and a little lithium. That is all the Big Bang produced. Carbon, oxygen, nitrogen, phosphorus, iron, all the atoms that life on Earth is built from, did not exist until the first massive stars exploded and forged them in their deaths, scattering them into space for the next generation of stars and planets to collect. That process took billions of years. But by about 10 billion years ago, the universe had enough of those elements available to build rocky planets like Earth. If any of those early rocky planets developed life, they had a head start on us of nearly 6 billion years. 6 billion years of evolution. Humanity has existed in its current form for roughly 300,000 years. Civilization, agriculture, writing, industry, for less than 10,000. Our entire technological history fits inside a sliver too thin to see on a cosmic timeline. A civilization with a 6 billion year head start would be so far beyond us that we genuinely cannot form a useful picture of what they could do. We cannot meaningfully imagine it any more than bacteria can imagine designing a telescope. If such a civilization exists anywhere in the observable universe, and if they have continued developing for that entire time, the universe should carry their marks. Not necessarily radio signals. Something far more fundamental: patterns in stellar distributions, large-scale structures. alterations to physics at scales we have barely begun to measure. Some scientists have proposed that certain large-scale features of the observable universe, structures that seem statistically unlikely to form by random processes, might be worth examining through this lens. The idea is speculative. It is not mainstream science, but it is discussed. What is mainstream science is the recognition that the window for life in the universe is nowhere near closing. The universe is not old. It is young, cosmically speaking. The stars that will burn longest have barely started, and around those long-burning stars, life might be getting its first foothold right now. Those stars have a name, and they are the most common type of star in the universe. They burn so slowly that they will outlast our sun by a margin too large to easily picture. And around some of them, planets have already been waiting in stable orbits for billions of years. Our sun will burn for about another 5 billion years before it begins to die. A red dwarf star will burn for up to 10 trillion years. 10 trillion. Our sun's lifespan, placed next to that number, is less than 1/10 of 1%. Red dwarfs are the tortoises of the stellar world. Small, cool, dim, and almost impossibly long-lived. They are also the most common type of star in the Milky Way. Roughly 70% of all stars in our galaxy are red dwarfs. Every direction you look in the night sky, the majority of stars you cannot see, because they are too faint to see without a telescope, are red dwarfs burning slowly in the dark. Planets have been found around red dwarfs. Several of them sit within the habitable zone, at distances where liquid water could theoretically sit on the surface. The most talked about case is a planet orbiting Proxima Centauri, the closest star to our Sun, just 4 light-years away. That planet sits in the habitable zone. It may have liquid water. The complications are real. Red dwarfs are temperamental. They produce frequent and violent flares, bursts of radiation, and high-energy particles that blast outward from the star. A planet close enough to be in the habitable zone is also close enough to take direct hits from these flares. That radiation could strip away an atmosphere or damage surface chemistry over time, but the timeline advantage is extraordinary. If a planet around a red dwarf can develop a thick enough atmosphere to shield its surface, and if life can get started there, it has trillions of years to develop. Earth got complex animal life only in the last 600 million years of its 3.8 billion year history of life. That is about 15% of the time available. A planet around a red dwarf with 10 trillion years available could cycle through hundreds of iterations of complex life, rising, going extinct, and new forms evolving in the aftermath. Each extinction event, like the ones that wiped out the dinosaurs and before them more than 90% of all species in the ocean, can also be an evolutionary reset, pushing new forms of complexity into the niches left empty. 10 trillion years is time for things to happen that we cannot begin to anticipate. Scientists are actively studying whether red dwarf planets can maintain atmospheres under flare conditions. Some models suggest thick enough atmospheres can survive and recover. Some models suggest the flares win. The answer depends heavily on the specific star, the specific planet, and the specific point in the star's history. If even a small fraction of red dwarf planets develop protected atmospheres and biology, the number of inhabited worlds in the galaxy is enormous. And some of those worlds are old enough to have civilizations that began before Earth even formed. What would scientists look for to find them? The answer to that question has changed completely in the last decade. The new search is quieter, more precise, and far more likely to actually work. Forget radio signals for a moment. The most likely way humanity finds alien life will not involve a dramatic broadcast cutting through the static. It will involve a graph, a spectrum, a set of numbers that, when scientists look at them carefully, do not add up without biology. Biosignatures are the chemical fingerprints that living systems leave in their environments. Every living thing consumes some chemicals and produces others, and those changes accumulate in ways that become detectable across planetary scales. Oxygen is the most obvious example. Earth's atmosphere is about 21% oxygen. That is extraordinarily unusual. Oxygen is chemically reactive. It combines with rocks, metals, and organic material constantly, pulling itself out of the atmosphere without a continuous replenishment source. Free oxygen in an atmosphere disappears within a few million years. On Earth, photosynthesis replaces it. Pull the life out of Earth's atmosphere, and the oxygen vanishes. A distant telescope detecting 21% oxygen in the atmosphere of an exoplanet would be looking at a planet that almost certainly hosts biology. No known geological or chemical process maintains free oxygen at that level without life. But oxygen alone is not enough. Scientists want to see combinations that make sense together only if biology is running them. Oxygen and methane existing simultaneously in an atmosphere is particularly significant. Methane breaks down quickly in the presence of oxygen. For both to coexist at detectable levels, something must be continuously producing the methane. On Earth, that something is living things, mostly microbes in swamps, wetlands, and animal digestive systems. Seasonal variations are another powerful signal. Earth's northern hemisphere has far more land than its southern hemisphere, and that land carries enormous amounts of vegetation. Every spring and summer, that vegetation absorbs carbon dioxide and releases oxygen. Every fall and winter, decomposition reverses the process. The result is a rhythmic rise and fall in atmospheric carbon dioxide that follows the calendar precisely. A telescope watching Earth from another star would detect this annual chemical heartbeat. It is a pattern that geology cannot produce. Only biology, responding to seasons, creates it. Scientists are also looking for the specific ratios of carbon isotopes that biology favors. Living things preferentially incorporate lighter carbon atoms into their structure, leaving a signature in the ratio between two forms of carbon that differs measurably from what purely chemical processes produce. Chlorophyll, the molecule plants use to absorb sunlight for photosynthesis, has a very specific spectral signature called the red edge. Vegetation absorbs most visible light but reflects a striking amount of near-infrared light. Seen from space, a planet covered in vegetation looks dramatically different in infrared than a rock-covered world. Scientists have already detected the red edge signature coming from Earth using instruments designed to mimic distant telescope observations. The same detection, aimed at an exoplanet, would be extraordinary. The James Webb Space Telescope is already collecting this kind of data. The instruments are not yet sensitive enough to pick up these signatures at the distances of known Earth-like planets. But the next generation of telescopes, currently being designed, will be. The search has shifted from hoping for a message to reading the chemistry of distant skies. And one planet in the James Webb data is already showing something scientists cannot fully explain. The planet is called K2-18b, and it sits about 120 light-years from Earth. 120 light-years is an enormous distance. Light traveling at nearly 600 billion miles per year takes 120 years to make that trip. The light James Webb collects from that planet's atmosphere left before anyone alive today was born. Despite that distance, the telescope can analyze the atmosphere in detail. When the planet passes in front of its star, a tiny fraction of the star's light filters through the planet's atmosphere before reaching James Webb's instruments. Different molecules absorb specific wavelengths of that light. By studying which wavelengths are missing from the star's spectrum, scientists can identify which molecules are present in the atmosphere. The planet is larger than Earth, but smaller than Neptune, a category scientists call a sub-Neptune. It orbits in the habitable zone of its red dwarf star. It may have a liquid water ocean covering its entire surface. Scientists call this type of world a Hycean planet, from the combination of hydrogen atmosphere and ocean. In 2023, the James Webb team published atmospheric data from K2-18b. They found carbon dioxide and methane, both consistent with a rich, chemically active atmosphere. Then they found something they had not expected, a tentative signal for a molecule called dimethyl sulfide. On Earth, dimethyl sulfide is produced almost exclusively by marine phytoplankton, microscopic ocean-dwelling life forms. It is released into Earth's atmosphere in enormous quantities by the oceans. It plays a role in cloud formation. It is detectable from space, and on Earth, it has no significant non-biological source. The team was careful. The signal was at the edge of detection. They used the word tentative specifically. The data required follow-up observations to confirm. Scientists outside the team immediately began discussing whether geological or purely chemical processes on an alien world with different chemistry than Earth might produce dimethyl sulfide without biology. The honest answer, as of now, is that the signal needs more data. Follow-up observations are scheduled. The confirmation or the refutation of that signal is one of the most anticipated results in current astronomy. But set the specific molecule aside. The broader point is that the James Webb Space Telescope is already doing what scientists dreamed about for decades. It is reading the chemistry of atmospheres around worlds orbiting other stars. It found carbon dioxide on an exoplanet. It found methane. It found water vapor. These are real detections, real data from planets light years away. The instrument is operational. The targets are identified. The question is no longer whether we can detect biosignatures across interstellar distances. The question is whether any of the atmospheric signals already in the data contain the answer we have been building toward for 60 years. Scientists watching that data come in, checking each new transmission from the telescope, know that the next detection might be the one. And if it is, the implications are not just scientific, they are existential. One microbe changes everything. That is the part that does not fit easily in the mind. A single, confirmed detection of life anywhere other than Earth, even something invisible to the naked eye, even something that does nothing more complex than eat a chemical and divide, would be the most significant discovery in human history by a margin that is difficult to overstate. Here is the mathematical logic that makes it so profound. Earth formed about 4.5 billion years ago. Life appeared here roughly 3.8 billion years ago, within 700 million years of the planet forming. The universe contains somewhere between 200 billion and 2 trillion galaxies. Each galaxy contains hundreds of billions of stars. A large fraction of those stars have planets. Conservative estimates suggest there are at least one followed by 24 zeros of habitable environments in the observable universe. If life on Earth arose from random chemistry within 700 million years, and Earth is a completely ordinary planet around a completely ordinary star in a completely ordinary part of the galaxy, then the probability of life forming on any given suitable world is some number. Call it any number at all. Multiply that number by the count of suitable environments. If life is even slightly probable given the right conditions, the result across that many environments is enormous. Billions of inhabited worlds, possibly trillions. The only way to escape that math is to argue that Earth is special in some unique and non-obvious way, that the precise conditions here are so extraordinary that they occur only once in the universe. Scientists call this the rare Earth hypothesis. It is a serious scientific position, but it requires Earth to be cosmically unique, a coincidence of conditions so improbable that no combination of factors has reproduced it anywhere else in the observable universe. Find one microbe on Mars, and rare Earth collapses. Find chemistry in the European Ocean that only biology explains, and rare Earth collapses. Detect dimethyl sulfide around a planet 120 light-years away that has no chemical explanation, and rare Earth collapses. What replaces it is a universe overflowing with life, common life. Microbial life distributed across billions of worlds, most of it never developing into anything complex, but present, persistent, a background condition of the cosmos rather than a rare accident. And if simple life is common, the question of complex life reopens with completely different odds. If a billion worlds carry microbes, Even if only 1 in a million of those ever develops multicellular life, that is still 1,000 civilizations in the Milky Way alone. What does that mean for us? Humanity has built its entire sense of identity around being special. Religious traditions, philosophical frameworks, artistic movements, the stories cultures tell about why we are here and what we mean, all of them, in different ways, assume that what happened on this planet matters in a cosmic sense. A universe full of life does not erase that meaning, but it transforms it. We are no longer the exception, we are an example, one among many, scattered across a living universe, each making something of the biology they were handed by the chemistry of their particular world. The search for alien life is, at its deepest level, the search for context, for an answer to the oldest question any conscious creature can ask: are we alone? The answer is coming. The telescopes are running. The data is flowing. The chemistry of distant skies is being decoded for the first time in history. Whatever that answer turns out to be, it will not leave us unchanged.